deep inside our own cells. All right, let's dive right in. We're going on a pretty incredible journey deep inside our own cells to understand the fundamental mechanics of, well, how we exist.
I mean, really think about it. We all start as one tiny cell, and somehow that becomes us. It's this amazing story of growth and repair, a process that's happening constantly, right now, inside your body. So how does it all work?
Well, the secret is this unbelievably elegant process. It's all about perfect packaging and precise duplication. Think of it as a masterclass in microscopic engineering that's running the show inside every one of us. Okay, let's get into it. To really get this, we first have to appreciate a truly mind-boggling puzzle of scale. A puzzle that, amazingly, your body has already solved.
2.2 meters. If you took the DNA from a single one of your cells and stretched it out, that's how long it would be. Yeah, that's taller than the average person. It's this massive instruction manual for building you.
So here's the big question. How in the world do you cram 2 meters of anything into a space that's thousands of times smaller? It sounds completely impossible, right? But our cells do it all the time.
The solution is this incredibly clever packaging system. It's kind of like molecular origami. Let's see exactly how this works.
So, for most of a cell's life, its DNA is in a form called chromatin. You can think of this as the working library. It's uncoiled and accessible, so the cell can read different genetic instructions as needed. But when it's time for the cell to divide, you can't just move a messy library. You have to pack it up. That's when the DNA condenses into these tight little packages called chromosomes, the moving boxes. This keeps everything neat and prevents the DNA from getting tangled or broken during the big move.
And this is the very first step of that packing process. The long DNA strand wraps around these little proteins called histones, kind of like thread around a spool. This whole unit, the DNA wrapped around the protein core, is called a nucleosome. It's the basic building block of that supercondensed chromosome.
Now get this, and this might surprise you. A chromosome is actually more protein than it is DNA. Those histone proteins make up about 60% of the mass. They're not just passive spools, they're the critical scaffolding that makes this whole amazing feat of condensation possible.
Okay, so a cell doesn't just randomly decide to divide. Oh no, it has to prepare. It has a whole to-do list to get through first. This prep phase is called interphase, and it's actually where the cell spends most of its life.
And here is that to-do list. First, in the G1 phase, the cell just grows. It does its normal job and bulks up. Then comes the really critical step, the S phase. S stands for synthesis, and this is when the cell makes a perfect copy of every single strand of its DNA. Finally, in the G2 phase, it does one last check to make sure everything is perfect before the big division.
Now this is a super important point. After that S phase, the cell still has 46 chromosomes. The number doesn't double. But now each of those chromosomes looks like an X. Why? Because it's made of 2 identical copies called sister chromatids that are stuck together in the middle. It's one chromosome just holding a perfect backup copy of itself.
All right, all the prep work is done. It's showtime. This next part is called mitosis. It's the main event, a beautifully choreographed dance where the cell's nucleus divides to make sure both new cells get a complete set of instructions.
First up, prophase. All that stringy chromatin DNA starts to supercoil into those thick, visible chromosomes we were talking about. And at the same time, the membrane around the nucleus just dissolves, basically clearing the stage for the action to begin.
Next is metaphase, and this is just a moment of pure biological elegance. All 46 of those X-shaped chromosomes line up perfectly, single file, right down the middle of the cell. It's like a perfectly synchronized drill team making sure the split is exactly even. And here we go, the most dramatic part, anaphase. The sister chromatids get pulled apart. They literally rip away from each other and are dragged to opposite ends of the cell. This is the move that ensures each new cell will get one complete copy of the genetic blueprint. And finally, telophase. The chromosomes arrive at their destinations at opposite poles. A new nuclear membrane forms around each group, and the chromosomes start to relax and uncoil back into that working chromatin form. And just like that, the division of the genetic material is complete.
But hold on, we're not totally done. We have 2 nuclei, but we still have one big cell. The rest of the cell, the cytoplasm, has to split.
In animal cells like ours, the membrane just pinches inward from the outside, like pulling the drawstring on a bag until it closes. But plant cells have a rigid wall, so they can't do that. Instead, they build a new wall, called a cell plate, right down the middle, separating the two new cells from the inside out.
So everything we just saw, mitosis, that's for growth and repair. It makes identical cells. But there's a whole other kind of cell division called meiosis, and its purpose is totally different. It's all about making the next generation.
Just look at the key differences here. Mitosis makes 2 identical cells, and they have the full set of 46 chromosomes. Meiosis, on the other hand, makes 4 cells, and they're all genetically unique. But the biggest deal? It halves the chromosome number down to 23. These are the sex cells, sperm and eggs. And you have to have the chromosomes, right? So that when they combine, the baby gets the correct total of 46.
So where does all that genetic uniqueness come from? From this amazing process called crossing over. During meiosis, the paired chromosomes you got from your parents, one from your mom, one from your dad, literally get tangled up and swap pieces of DNA. This gene shuffling is exactly why you and your siblings aren't identical clones. It's the engine of genetic diversity.
Okay, so let's zoom back out for a second and just think about what this all means, what this incredible microscopic dance really signifies.
Every single moment of your life, every time a cut heals, every time you grow, every single day as old cells are replaced, all of it is powered by this precise, elegant, and constantly repeating cycle. It's a system that has been working non-stop inside you since the very beginning.
And that brings us to one last thought. This whole process is so incredibly accurate, it's almost perfect. But what happens on that rare occasion when it isn't? When a mistake slips through? Well, those rare errors, they're the source of a whole host of other biological stories. And that is a topic for another day.